Bamboo has innumerable applications in the modern era. However, the traditional propagation through vegetative methods like offset, rhizome, culm cuttings, branch cuttings, and layering are not sufficient to meet the market demand of quality planting material. Institute of Wood Science and Technology, Bengaluru has developed micropropagation techniques through plant tissue culture, which is proven to be a better alternative to the conventional modes. It helps to generate planting stock in large quantities. In vitro axillary shoot proliferation. Step 1. Explant collection. Nodal segments are collected from newly emerging shoots from healthy, matured culms. Step 2. Surface sterilization. The surface of the nodal segments are swabbed with ethanol. The explants are then washed with fungicide and antibiotic, followed by rinsing with distilled water. The explants are surface sterilized with 70% ethanol, followed by rinsing with autoclave distilled water. It is then washed with mercury chloride and rinsed thoroughly with autoclave distilled water. Step 3. Inoculation. The nodal shoot segments are inoculated upright in MS liquid medium for shoot induction. The cultures are incubated in growth chamber at around 25-degree temperature and 2500 lux intensity of light for a photo period of 12 hours. Step 4. Shoot multiplication. After sprouting of shoots, shoot clumps are transferred from the shoot induction medium to fresh MS liquid or agar gelled medium for shoot multiplication. Multiplied shoot clumps are regularly subcultured and transferred to fresh shoot multiplication medium. Step 5. Rooting. Clumps of 3 to 4 shoots are then transferred to agar gelled MS medium for in vitro rooting. Step 6. Hardening. Well-developed rooted shoots produced in vitro are carefully taken from the culture bottles and washed with water to remove the adhering medium. The plants are then washed in fungicide solution as a prophylactic measure against fungal attack. They are transferred to containers containing sand and kept inside polyglobules in the mist chamber for 2 to 3 weeks for primary hardening. The relative humidity and temperature are maintained. The plants are transplanted to polybags containing potting mixture and kept inside the mist chamber for 2 to 3 weeks for secondary hardening. And then the plants are kept under shade for 2 weeks before transferring to the open nursery. Tissue culture technology offers large-scale multiplication of elite clumps for large-scale plantation of bamboos. At IWST, complete micropropagation protocol has been developed for 15 bamboo species. The Institute transfers the protocols developed and also supplies in vitro-based cultures in bottles and hardened plantlets of these bamboo species to the stakeholders including the forest departments, private tissue culture establishments and farmers. For any consultation, training, information and assistance, contact Plant Tissue Culture Lab, the Institute of Wood Science and Technology, Bengaluru.